Welcome all to our channel Sri Raj Lakshmi Publication. Agriculture is the backbone of the Indian economy. Indian farmers are cultivating their farms in traditional way, but nowadays farmers are adopting new techniques and others experiences in their farm. The state agriculture universities have a giant share in agricultural development as well as economic and social upliftment of the farmers. The state agriculture universities also play a vital role in nation building. Being a well-wisher of the students and the beneficiaries of these subjects, Sri Raj Lakshmi Publication has now decided to publish these subjects in video form on the basis of agricultural development, that is, agronomy, genetics and plant breeding, agri-entomology, horticulture, agricultural economics, agri-extension education, agri-engineering, plant pathology, soil science and agri-chemistry, animal husbandry and dairy science. I express my sincere thanks to all the respected authors of books, research papers, review articles, as well as e resources whose reference has been taken for these subjects and video who have contributed to this publication, and also to the students who refer these books and videos to develop their knowledge updated for their bright future. Suggestions for improvement of this video are welcome. Please comment, like, share and subscribe our channel. That is Sri Raj Lakshmi Publication. Sri Raj Lakshmi Prakashan publish a textbook on Introductory Microbiology. Author Dr. S. A. Raut. Chapter 1st, Introduction to Microbiology. Microbiology often has been defined as the study of organisms and agents too small to be seen clearly by the unaided eye, that is, the study of microorganisms. Agricultural microbiology is defined as a branch of microbiology deals with plant associated microbes and plant and animal diseases. It also deals with the microbiology of soil fertility, such as microbial degradation of organic matter and soil nutrient transformations. Scope and importance of microbiology in agriculture. Microorganism plays a very important role in agriculture because the soil microbes, bacteria and fungi are essential for decomposing organic matter and recycling old plant material. Some soil bacteria and fungi form relationships with plant roots that provide important nutrients like nitrogen or phosphorus. Although soil organisms comprise less than 1% of the total mass of a soil, they have a vital role in supporting all plants and thus animals. Some of their vital functions are described below. Number 1. Soil microbes break down organic matter. Microorganisms play an important role in the decomposition of organic matter. Different types of microbes are specialized to different types of organic matter, between them covering just about everything. Cellulose is degraded by bacteria, especially those of genus Cytophaga and other genera Bacillus, Pseudomonas, Cellulomonas, and Vibrio. A bacterium and fungal genera Aspergillus, Penicillium, Trichoderma, Cactomium, Crullaria. Lignins and proteins are partially digested by fungi, protozoa, and nematodes. Proteins are degraded to individual amino acids mainly by fungi, actinomycetes, and Clostridium. Under anaerobic conditions of waterlogged soils, methane are mainly carbon-containing product which is produced by the bacterial genera strict anaerobes, Methanococcus, Methanobacterium, and Methanosardina. Number 2. Soil microbes help to recycle nutrients. Soil microbes play a crucial role in returning nutrients to their mineral forms which plants can take up again. This process is known as mineralization. Number 3. Soil microbes create humus. When the soil microbes have broken down all they can, what's left is called humus, a dark brown jelly-like substance that can remain unchanged in the soil for potentially millennia. Humus helps the soil to retain moisture and encourages the formation of soil structure. Humus is also suspected of suppressing plant diseases. Number 4. Soil microbes create soil structure. Some soil microbes secrete polysaccharides, gums, and glycoproteins which glue soil minerals together, forming the basis for soil structure. Fungal hyphae and plant roots further bind soil aggregates together. Soil structure is essential to good plant growth. Examples are fungi like Rhizopus, mucor, Chaetomium, Cladosporium, Aspergillus, Trichoderma, and bacteria like Azotobacter, Rhizobium, Bacillus, etc. Number 5. Soil microbes fix nitrogen. Agriculture depends heavily on the ability of certain microbes, mainly bacteria, to convert atmospheric nitrogen into gas to ammonia NH3. Some live freely in the soil while others live in association with plant roots. The best example is Rhizobium bacteria in the roots of legumes. The process of conversion is known as nitrogen fixation. Biological nitrogen fixation contributes about 60% of the nitrogen fix on Earth. In contrast, manufactured fertilizers contribute 25%. As the cost of energy continues to rise, so too the cost of manufactured nitrogen fertilizers will rise. So biological nitrogen fixation is likely to have ever increasing importance in food production. The numbers of soil microorganisms carry out the process of biological nitrogen fixation at normal atmospheric pressure, 1 atmosphere, and temperature around 20 degrees Celsius. Two groups of microorganisms are involved in the process of BNF: A. Non-symbiotic free living and B. Symbiotic associative. Non-symbiotic free living. Depending upon the presence or absence of oxygen, non-symbiotic N2 fixation prokaryotic organisms may be aerobic heterotrophs, Azotobacter, Pseudomonas, Achromobacter, or aerobic autotrophs, Nostoc, Anabaena, Calothrix, BGA, and anaerobic heterotrophs, Clostridium, Klebsiella, Desulfovibrio, or anaerobic autotrophs, Chlorobium, Chromatium, Rhodospirillum, Methanobacterium, etc. Symbiotic, associative. The organisms involved are Rhizobium, Bradyrhizobium in legumes, aerobic, Azopirillum grasses, actinoncytes, practic with Cassorinus alder. Number 6. Soil organisms promote plant growth. Some soil microbes produce a variety of substances that promote plant growth, including auxins, gibberellins, and antibiotics. Number 7. Soil microbes as biocontrol agents. Several eco-friendly bioformulations of microbial origin are used in agriculture for the effective management of plant diseases, insect pests, weeds, etc. Example, Trichoderma sp are used for biological control of seed and soil-borne diseases. Fungal genera Entomophthora, Beauveria, Metarhizium, and Protozoa, Maltesia grandis, Malameba, Locustia, etc. are used in the management of insect pests. Nuclear polyhydrosis virus (NPV) is used for the control of Heliothis, American bollworm. Bacteria like Bacillus thuringiensis and Pseudomonas are used in cotton against bollworms and angular leaf spot respectively. Number 8. Bioinsecticides. Some beneficial microorganisms can also be helpful in developing alternative controls to synthetic insecticides. Formulas for coatings on the seed inoculants which carry these beneficial organisms can be developed to protect the plant during the critical seedling stage against insect pest attack. Bioinsecticides do not persist long in the environment and have shorter shelf lives. They are effective in small quantities, safer to humans and animals compared to synthetic insecticides. They are very specific, often affecting only a single species of insect and have a very specific mode of action. Slow in action and the timing of their application is relatively critical. Point A. Fungal bioinsecticides. Fungi cause diseases in some 200 different insects, and this disease producing traits of fungi is being used as bioinsecticides. Fermentation technology is used to mass production of fungi. Spores are harvested and packaged, so these are applied to insect-ridden fields. When the spores are applied, they use enzymes to break through the outer surface of the insect's bodies. Once inside, they begin to grow and eventually cause death. Example: Metarhism, Beauveria bassiana, etc. Fungal agents are recommended by some researchers as having the best potential for long-term insect control. This is because these bioinsecticides attack in a variety of ways at once, making it very difficult for insects to develop resistance. Point B. Virus-based bioinsecticides. Baculoviruses affect insect species like corn borers, potato beetles, flea beetles, and aphids. One particular strain is being used as a control agent for Bertha army worms, which attack canola, flax, and vegetable crops. Traditional insecticides do not affect the worm until after it has reached this stage, and by then much of the damage has been done. Number 9. Degradation of pesticides in soil by microorganisms. Soil receives different toxic chemicals in various forms and causes adverse effects on beneficial soil microflora, microfauna, plants, animals, and human beings. Various microbes present in soil act as the scavengers of these harmful chemicals in soil. The pesticides chemicals reaching the soil are acted upon by several physical, chemical, and biological forces exerted by microbes in the soil, and they are degraded into non-toxic substances and thereby minimize the damage caused by the pesticides to the ecosystem. For example, bacterial genera like Pseudomonas, Clostridium, Bacillus, Theobacillus, Achromobacter, etc., and fungal genera like Trichoderma, Penicillium, Aspergillus, Rhizopus, and Fusarium are playing important role in the degradation of the toxic chemicals pesticides in soil. Number 10. Biodegradation of hydrocarbons. Natural hydrocarbons in soil like waxes, paraffins, oils, etc. are degraded by fungi, bacteria, and actinomycetes. Example: ethane, C2H6, a paraffin hydrocarbon, is metabolized and degraded by mycobacteria, nocardia, streptomyces, pseudomonas, flavobacterium, and several fungi. Number 11. Microbiology, ecology, biotechnology, and sustainable agriculture. An increased knowledge of microbe based symbiosis in plants could provide effective ways of developing sustainable agriculture in order to ensure human and animal food production with a minimal disturbance of the environment. The effective management of symbiotic microbial communities is possible using molecular approaches based on the continuity of microbial pools which are circulating regularly between soil, plant, and animal provided needs in natural and agricultural ecosystems. Kuprianov et al. 2010 analysis of this circulation could enable the creation of highly productive microbe-based sustainable agricultural system. Number 12. Environmental health and microbial biotechnology. With recent advances in biology, materials, computing, and engineering, environmental biotechnologists now are able to use microbial communities for a wealth of services to society. These include detoxifying contaminated water, wastewater, sludge, sediment, or soil, capturing renewable energy from biomass, sensing contaminants or pathogens, and protecting the public from dangerous exposure to pathogens. Number 13. Bioherbicides. Weeds are the problem for farmers. They not only compete with crops for water, nutrients, sunlight, and space, but also harbor insect and disease pests. Clog irrigation and drainage systems, deteriorate crop quality, and deposit weed seeds into crop harvest. Bioherbicides are another way of controlling weeds without environmental hazards posed by synthetic herbicides. The microbes possess invasive genes that can attack the defense genes of the weeds, thereby killing it. The benefit of using bioherbicides is that it can survive in the environment long enough for the next growing season where there will be more weeds to infect. It is cheaper than synthetic pesticides, thus could essentially reduce farming expenses if managed properly. Further, it is not harmful to the environment compared to conventional herbicides and will not affect non-target organisms. Number 14. Deriving from various naturally occurring microorganisms such as bacteria and fungi, microbial technologies can protect crops from pests and diseases and enhance plant productivity and fertility. They enable farmers to increase yield and productivity in a sustainable way and are expected to play a significant role in agriculture. Number 15. Microbial solutions enable farmers to drive yield and productivity in a sustainable way. Deriving from various naturally occurring microorganisms such as bacteria and fungi, these solutions can protect crops from pests and diseases and enhance plant productivity and fertility. Microbial solutions provide more choice for farmers and help meet the demand for more sustainable agricultural practices. Such solutions can increase crop yields and develop a more sustainable industry impact profile, ultimately resulting in more food to feed the growing world and new opportunities to protect the planet. Number 16. Microbial technologies can help to improve nutrient acquisition, promote growth and yield, control insects, and protect against disease. These emerging agricultural biological technologies complement the integrated systems approach that is necessary in modern agriculture, bringing together breeding, biotechnology, and agronomic practices to improve and protect crop yields. Branches of microbiology. The branches of microbiology can be classified into pure and applied sciences. Microbiology can be also classified based on taxonomy in the cases of bacteriology, mycology, protozoology, and phycology. There is considerable overlap between the specific branches of microbiology with each other and with other disciplines, and certain aspects of these branches can extend beyond the traditional scope of microbiology. Point A. Pure branches of microbiology. Subpoint 1. Based on taxonomic arrangement. Bacteriology, the study of bacteria. Mycology, the study of fungi. Protozoology, the study of protozoa. Phycology or algology, the study of algae. Parasitology, the study of parasites. Immunology, the study of the immune system. Virology, the study of viruses. Nematology, the study of the nematodes. Subpoint 2. Integrative arrangement. Microbial cytology. The study of microscopic and submicroscopic details of microorganisms. Microbial physiology. The study of how the microbial cell functions biochemically, includes the study of microbial growth, microbial metabolism, and microbial cell structure. Microbial ecology, the relationship between microorganisms and their environment. Microbial genetics, the study of how genes are organized and regulated in microbes in relation to their cellular functions, closely related to the field of molecular biology. Cellular microbiology, a discipline bridging microbiology and cell biology. Evolutionary microbiology, the study of the evolution of microbes. This field can be subdivided into microbial taxonomy, the naming and classification of microorganisms. Microbial systematics, the study of the diversity and genetic relationship of microorganisms. Generation microbiology, the study of those microorganisms that have the same characters as their parents. Systems microbiology, a discipline bridging systems biology and microbiology. Molecular microbiology, the study of the molecular principles of the physiological processes in microorganisms. Point B. Other branches of microbiology. Nanomicrobiology, the study of those microorganisms on nano level. Exomicrobiology or astromicrobiology, the study of microorganisms in outer space. Biological agent, the study of those microorganisms which are being used in weapon industries. Point C. Applied branches of microbiology. Medical microbiology, the study of the pathogenic microbes and the role of microbes in human illness, includes the study of microbial pathogenesis and epidemiology and is related to the study of disease pathology and immunology. Pharmaceutical microbiology, the study of microorganisms that are related to the production of antibiotics, enzymes, vitamins, vaccines, and other pharmaceutical products and that cause pharmaceutical contamination and spoil. Industrial microbiology, the exploitation of microbes for use in industrial processes. Examples include industrial fermentation and wastewater treatment, closely linked to the biotechnology industry. This field also includes brewing, an important application of microbiology. Microbial biotechnology, the manipulation of microorganisms at the genetic and molecular level to generate useful products. Food microbiology and dairy microbiology, the study of microorganisms causing food spoilage and foodborne illness, using microorganisms to produce foods, for example, by fermentation. Agricultural microbiology, the study of agriculturally relevant microorganisms. This field can be further classified into the following: plant microbiology and plant pathology, the study of the interactions between microorganisms and plant pathogens. Soil microbiology, the study of these microorganisms that are found in soil. Veterinary microbiology, the study of the role of microbes in veterinary medicine or animal taxonomy. Environmental microbiology, the study of the function and diversity of microbes in their natural environments. This involves the characterization of key bacterial habitats such as the rhizosphere and phyllosphere, soil and groundwater ecosystem, open oceans or extreme environments, extremophiles. This field includes other branches of microbiology such as microbial ecology, microbially mediated nutrient cycling, geomicrobiology, microbial diversity, bioremediation, water microbiology or aquatic microbiology, the study of those microorganisms that are found in water, aeromicrobiology or air microbiology, the study of airborne microorganisms. And for next chapter, please don't forget to comment, share and subscribe our channel, that is Sri Raj Lakshmi Publication. Thank you.